Good morning, students. Please inform your friends. Let them join as early as possible. YouTube Live and Facebook Live is also going on. Please check it. Who had not joined, let them join. Okay. Within 2 minutes, Sir will join and section will be start. Please check it. Please inform to your friends who has not joined. Let them join as early as possible. YouTube Live and Facebook Live is also going on. Please check it there also. You can watch there also. Good morning, students. Please let them join as early as possible. Within 2 minutes, Sir will join and the section will be start. Please check it. YouTube Live and Facebook Live is also going on. You can watch there also.
हाथ लगे ना ओके थोड़ा पानी अच्छा एक बॉटल आपी देजो મને અહીંયા બાજુ મૂકી દેજો બોટલ हेलो
Good morning, sir.
very good morning Maheshwari uh, yes I'm all set whenever you are ready just let me know
Yeah, ready. You can continue, sir.
all right so very good morning to all of you is my voice audible
Yes, sir.
okay great let me just share my screen and then we get started
Okay, sir. Okay, sir.
are you able to see my screens
Yes, sir.
fantastic all right so let's just go with the
Sir, please increase your brightness in the laptop because it is blur. There is no clarity.
on my video or on the uh,
PPT. All
on the
you have to.
yeah PPT. One second, I'm yet to just 1 second. It will be okay. Just 1 second. Yes, are you able to see now? Maheshwari, are you there?
Yes, sir. 1 minute, sir.
Yeah, yeah,
Your screen. Yeah.
yeah. Are you able to see?
Just a minute, sir. It is
Yes.
blur.
It is still blurred. This is so strange.
Yeah.
Is it okay now?
1 minute, sir. Yeah, okay.
All right. Okay, great. So very good morning to all of you, and we will start from where we stopped last time. That is essentially about the models we were talking. So very quickly, we'll have a recap of what we have discussed in the last class about the hepatocytes and enterocytes. And their difference between hepatocyte, enterocytes, their usage, their application part, and then we will talk further about ADME, in vitro assays today. Okay, so we talked about animal experimentations, requirement of animal experimentation without doing experimentation or testing in animals. Can we really do drug discovery and development? That was a question we discussed, and yes, uh, yes and no, because yes, because we cannot. I mean, we can definitely reduce the indiscriminate usage of animals, but we cannot really avoid completely animal usage in drug discovery and development. So that's why I say yes and no. Are there any better alternatives? I mean, how do you really reduce the usage of animals in drug discovery and development research? That is by using alternate models of cell-based models, right? 2D and 3D cultures, cell cultures can be used, which can really be one screening tier for screening of your molecules. And then we talked about that as on date, even in India, as in Western countries, the drug discovery and development approach is it's starting from in silico screening of molecules of, I mean, in-house libraries, followed by in vitro experimentations in lab, and then finally we go to testing in live animals, which could be any of the laboratory animal models of your disease target based on the disease target you have selected. Could be rats, mice, guinea pig, hamster, rabbits, or legal dogs. So we talked about why experimental design is required, because all the animals, laboratory animals or larger animals or even human, we are different. Physiologically, we are different. Our genetic makeups are different. In surface area availability, also we all are different. So that's why we have to twitch the doses according to the species we are using. So the extrapolation of dose from smaller species to higher, or extrapolation of the data, we call this as allometric scaling. And in a separate class, we'll be covering about allometric scaling in detail. So then we talked about hepatocytes and enterocytes, and both of them are novel. Of course, hepatic technology is not novel. It is there in public domain for almost more than 30-35 years. And enterocytes have come very recently because of our increased understanding on metabolism. I think scientists across globe have realized that first site of metabolism is in intestine, and intestine is one of the first organs which is going to see your drug. So after it gets absorbed, if there is an expression of enzymes, metabolizing enzymes in intestine, in enteric cells, then metabolism can happen there itself. So that's why enteric technology is definitely new. We have now understood completely about the fate of your drug. Now this is following oral administration. If you take a dose, a pill, say uh, any anti-inflammatory drug, any drug, any tablet, drug, pill you're taking orally, say brufen or paracetamol or cetirizine, after it is taken up with water, it goes to through esophagus, it reaches stomach. From stomach, it will go to intestine, and that's where it will get absorbed. And once it gets absorbed, it will cross the lipid bilayer membrane of intestine. From there, through portal circulation, it goes to liver. In liver, there will be metabolism, and after metabolism, and of course, first site of metabolism, as we have understood, would be intestine, and then second site would be liver. After that, drug would be distributed across the body, and then it goes to different tissues. So binding of drug can happen to proteins in the tissues also. So that would be non-specific binding. Even in blood, there are plasma proteins to which your drug can get bound. So that would be the bound form of your drug. And to elicit a pharmacological action, you need free drug. So that free portion of drug is the only drug concentration which is responsible for eliciting a pharmacological action. So it can bind to the ligand, it can bind to the receptors based on the mechanism of your drug action. And once metabolism happens, then the drug becomes more polar. I mean, the metabolites are polar, or even as such drug, if it doesn't get metabolized also, it will have an opportunity to go to kidney, and through kidney, it will be eliminated from your body. So this is the whole area of absorption, distribution, metabolism, and excretion. On the then we also talked about on this slide, as you can see, that there are expressions of cytochrome P450 enzymes in intestine as well. So 3A4 and other isozymes 2A2, 2A1, 2B6, 2D6, all of them are expressed in intestine also. Once through portal vein they reach to liver. In liver also same cytochrome P450 enzymes are expressed. So metabolism can happen there also. So for bioavailability, final bioavailability of your drug, metabolism in intestine, in enteric cells as well as in hepatic cell is contributing. Expression of enzymes, we already talked, 3A4 widely expressed in intestine. Same way in liver also, 3A4 and other isozymes are expressed in abdomen, which are responsible for metabolism of your drug. And of course, there are transporters also, efflux and uptake transporters. Uptake transporters will help in taking the drug inside the cell, and efflux transporters will avoid the entry of your drug into the cell. So if efflux transporters are expressed in intestine, which is majorly PGP substrate, if your drug is a PGP substrate, then the bioavailability will be less, because every time the drug tries to cross this barrier, efflux transporters, PGP transporters will take your drug back into the intestine. So so to understand the role of transporter is also equally important. Then we talked about hepatocytes. We already talked that it is considered gold standard for in vitro metabolism studies, and also to study drug-drug interaction, and in doing hepatotoxicity study also, these are important. And why we call it as gold standard, why they are highly studied, researched cell model, because they got intact plasma membranes, and they have uninterrupted enzyme levels. All the enzymes are expressed in that, and also there is expression of transporters. So once you study the metabolism in hepatocytes, it will not only tell you metabolism, but in case if your drug is a PGP substrate, even that can, you will get an idea about it based on hepatocyte data which you get. So we'll come to know that whether there will be any issues related to transporters while our drug tries to cross the intestinal barrier. So that's why hepatocytes are considered gold standard. We always use cryopreserved hepatocytes. The reason being, you can schedule your experiment, you can repeat the experiment, you can use actually the pre-characterized hepatocytes, where suppose if we want to study, we want a lot of hepatocytes where there is more expression of 3A4, then you can get pre-characterized hepatocytes available from the market. There are vendors who are supplying this, especially one such vendor about that data which we are going to share is from IVL in vitro admet labs. So they're widely used hepatocytes across country. So you can use pre-characterized hepatocytes. Then not only from one single donor, suppose we want to study the variability, like sometimes students have questions that you can study all this thing only in few individuals, whereas when the drug is launched in the market, it is going to be utilized by a larger section of population. So how do you really correlate that? So to understand that variability, instead of studying in single donor hepatocytes, that is from one individual, we can use multiple donors together. So suppose 10 human livers have been mixed together, and we have a pooled hepatocytes from that. So if there are any variabilities, we can that can reflect in your metabolism experiment also. Then we talked about UCRM, that once you have your hepatocytes, your cells, enterocytes or hepatocytes in cryopreserved form, while they are in cryopreserved form, they are actually at minus 170 or 180 degrees in liquid nitrogen. Once you take it out and you thaw them because you want to use it, then to recover that, there is a media which is called universal cryopreserved recovery media, cryopreservation recovery medium, right? And I was talking about pre-characterized. So as you can see, each lot from any vendor you buy, each lot of hepatocyte will have all this data available. That the age of the person from whom it is collected, the gender, race, yield, and viability. For hepatocytes, the ideal yield is between 6 to 7 or 8 million cells per vial. Viability, when you're talking of hepatocyte, we always talk of more than 90% viability. So where now we talked about hepatocytes, cryopreservation. Now there is something called as platability. Means platable lots are available. Of course, you can have even today you can use it in a suspension form, but there are platable lots which are available. So in suspension, would, as you can see, they rapidly lose the viability. Like within 6 hours, the viability would be half. From 90% it will come down to almost 45 or 50%. So there's therefore only short-term experiments where your incubations are only for few hours you can do using suspension form of hepatocytes. Whereas plated hepatocytes, you can do long term because they are viable for days and sometimes for weeks, up to 3 weeks. Also, they can be viable. So, if you are doing long term incubations, you can use platable lots. You can use for hepatotoxicity, even metabolism dependent drug toxicity, and enzyme induction inhibition studies and efflux transporter studies. All these studies are long term studies where you need incubation more than a day. So that's where platable lot of hepatocyte is preferred, that you should use those. This is how they actually look. Colonies, you will see hepatocytes from human, dog, mouse, and Wistar rats. Then they are all the slots are being characterized. So this is just the data we have shared about induction of 1A2 using HH1083. HH is human and 1083 is the lot number. So this particular lot of hepatocytes in that induction of CYP1A2 in presence of omeprazole, phenacetin, and rifampicin. So you see 2B6 with all these three, you're seeing dose dependent induction, right? This is relative expression. Here again, with omeprazole, phenacetin and all, for 3A4, for the same lot, there is a dose-dependent increase in the expression of the enzyme. So these are highly inducible lot. So we talked about the viability, more than 90% for hepatocytes, almost confluent if they are platable. They do express 1A2, 2B6, 3A4, intact uptake and efflux transporters activity are there, and for long duration you can use over 7 days in culture you can use it. So that's why these are preferred. There are different species available, like you can do it from human, mouse, rat, beagle dog, cynomolgus monkey, and or rhesus monkey. So what happens is from small to large entire species you have a range of cells, hepatocytes from all the species, and we can compare the data, that metabolism, how does it look like in rats, in mice, in dog, or in monkeys. Somebody other day also asked a question that there is a difference between animal to human genome. Okay, so not that is very much true. Human and animals are completely different. But the question here is that can we really go ahead and test every drug in human directly? Answer is no, it's not possible. Of course, from ethical point of view, but also once your drug is under discovery stage, you this is not a final product, so you don't know what to expect in terms of side effects, in terms of efficacy. So that's what we are screening the molecules in preclinical for, right? So if at all I have to test directly in human, if it was doable, then yes, always I would like to test in human, but that's not possible. So, like thousands of molecules, all of them cannot be tested in human. So that's why to filter, to have some barrier there, we have started doing screening trials in lower species. Of course, genomes are different, but this all genomes are well known. I mean, the expression of enzymes Cytochrome P450 enzymes in mouse is known, in rats it is known, in beagle dog it is known, even in monkeys and human it is known. The expressions are all known. Suppose 3A4 is expressed in mouse, same 3A4 in beagle could be 3A1, but that is known. So we already know the comparison. And at the same time, this is a very good point, a good question, because whenever you get any metabolism data from this lower species, any species, rat, mice, dog, monkey, we should always be careful when we interpret the data. We have to see if the metabolism pattern is going to be same in human. So if those expression or those enzymes which are expressed in this species, and if you do a reaction phenotyping study, you'll know which particular isozyme is responsible for metabolism in rat, in mouse. You should know the equivalent form of that enzyme or same enzyme, does it also express in human? If it doesn't, then the metabolism data which you get in mouse and rat may not be the data which you will expect in human. So all these factors are taken into consideration when you actually interpret the data. So yes, to answer the question, yes, both the genomes are different. The expression of enzymes in liver and intestine could be different, but you have to keep that variability in mind, and then you have to interpret the data. Okay, and that's the only reason many a times drugs are, say, highly metabolism, they are undergoing high metabolism via in rats and mice, whereas when in human they are rock solid, they are completely stable. Why? Because the expression of enzymes is different. So that you have to understand while you interpret the data. Still, in spite of having all these issues with it, still it is useful to use this metabolism in lower species as screening tier for our compound, so that only fewer compound can proceed further. I hope I have answered that question. So there are new hepatocyte pools. I already talked. We can check cryopreservation, cryopreserved human hepatocytes, and there are something called as quick refreeze. So cryopreserved were thawed. Again, they were cryopreserved. So from multiple donors, also it can be pooled after they have been cryopreserved. So that technique is patented. This is the US patent number. Even that is doable. So we have pooled suspension human hepatocytes. Again, just to address the variability issue, when we go for a larger population, we prefer to have pooled lot of hepatocytes for doing be it suspension or a platable, but we can use pooled suspension or platable hepatocytes in order to address the variability in the data. So applications of multiple donors, so you can check metabolic stability, metabolite profiling, reaction phenotyping, which particular isozyme is responsible for metabolism. You can do a affinity of your drug is towards are they really substrate for an uptake transporter or an efflux transporter? If they are a substrate for uptake transporter, more and more drug can be delivered inside the cell. If they are a substrate of efflux like a PGP substrate, then it cannot cross the barrier and it will be delivered back into the intestine, or even inside the brain. Also, blood-brain barrier, there are expressions of this transporters. So if your drug has to reach inside the brain, and if they are PGP substrates, even from there also drug can be delivered back into the blood rather than crossing the blood-brain barrier. Yeah. So then we do inhibition studies, like if our drug is responsible for inhibition of any of the enzymes in liver, those studies are called P450 inhibition. Same way, are they inducible? So that is P450 induction. And then finally we can study the hepatotoxicity. Then we talked about enterocytes. As we already discussed this, enterocytes are actually they are the first organs, one of the first organs which sees your drug first. So there is an expression of 3A4 majorly, predominantly they are expressed in intestine. So in enteric cells also it is equally important to study the metabolism, and we call this as first pass effect, first pass metabolism before liver. Okay, so intestinal drug-drug interaction, and as you can understand, within the intestine there could be a food material also available. So interaction of our drug with the food also can be studied if you have enterocytes as model cell model available with you. As of now, not many companies supply. IVL, for sure, I know they have a commercial model of enterocytes to study the metabolism. So let's talk about morphology. As you can see, these are smaller cells compared to hepatocytes. In terms of protein content, hepatocytes have more protein, like 1.45 mg per million cells, whereas in enterocytes, it comes to somewhere around 0.2 mg protein only. In terms of yield and viability, again, it is less than hepatocytes. Still, it's a good model to study. How it is less? So viability in enterocytes would be anywhere around 80% or even 75%, and yield would be less than 2 million cells per vial. So this is the characteristics of enterocytes. Again, they are also characterized, all the data of the lot is available, which gender, race, age, the body mass index, every data is available from the individual from where the enterocytes have been taken. Let's talk about say specific markers. How do you know if somebody just gives you a cell, and if you can't make out using morphology which particular cell type it is, then you should look for expression of cell-specific markers. So for enterocytes, there are two. One is sucrose isomaltase (SI) and maltose glucoamylase, which is MAGAM. And for hepatocytes, it will be showing the expression of albumin. So this is cell type markers expression we have seen. If the cells are unknown to you, if you see if you do the gene expression studies, you will see sucrose isomaltase and MAGM expression more if it is enterocytes, and you will see albumin expression more if it is hepatocytes. And they are very specific. They don't express in enterocyte at all. So it's yes or no decision to you. You can easily make out what cell type it is. Then this we already talked. Major expression of enzyme. The major enzyme which is expressing in intestine and liver is CYP3A4. And please remember, CYP3A4 is responsible for metabolism of more than 75% of the drugs which are available in the market. So, as you can see in intestine, out of all the enzymes expressed, 82% is only 3A4, right? 3 enzyme. Same way, in liver it is 40%. That's why if at all your drug is metabolized by 3A4, then it would definitely make sense to study that for intestinal metabolism. Again, expression of all the enzymes, isozymes like 1A1, 2B6, 2C8, 2C9, 2C19, difference between enterocyte and hepatocytes. Almost all are expressed in both, like 1A2 is very specific to liver. It doesn't express us in enterocytes. Same way, like 3A5 is lesser in enterocytes, but it is highly expressed here. But there is one enzyme 2S1 which is predominantly expressed only in enterocyte, but it doesn't express us in liver. This is a paper where it is already published 2S1 expression in in enterocytes. Viability is up to 75% or 80% and 1 to 3 million cells per vial for enterocytes. And then we also talked about the usage, where exactly they are applicable. So they can be used for enteric metabolic stability, metabolite identification, drug-drug interaction, drug-food interaction, and then finally doing enteric toxicity. So then we got into, I think this is where we stopped last time. So we'll continue from here. So within hepatocytes, what is new? So this is a new thing which has come up. It is commercially available. It is called 999 elite cryopreserved human hepatocytes. Why they are elite? But as the name suggests, they have more than 90% viability. They have more than 90% confluency, and for more than 9 days, these cultures can maintain this characteristic. So for all kind of long-term metabolism studies, this is the best lot of hepatocytes to be used, called as 999 elite. So there is another newer technology called as MatMax. Okay, so MatMax hepatocytes is nothing but they are permeabilized. So as you can see, a slight difference in the cells, because here they are all permeabilized, right? And versus their pores being made on the cell, so they are permeable. And now through those pores, we have supplied them with more of a cofactor, so that these are highly activated hepatocytes. So they will have always higher metabolism potential. Of course, this is not a normal scenario because normally hepatocytes are intact, right? In a human cells also or an animal cell, they are intact. But we have made this thing permeabilize so that more of the cofactor can get inside. And the advantage is now even the smallest metabolism, if it has to happen, can be visible using MetMax. Again, I'm saying it is commercially available, but here the purpose is not to study the normal metabolism, but sometimes we may miss out if metabolism is happening at a very low level. Even for those compounds, MetMax screening would really help. We can't miss out any metabolite if we use MetMax. Okay, this is the pictorial presentation. Generally, animal or a human hepatocytes, they are intact, whereas this metmax, the vendor has made it porous so that more of the cofactors can be added from outside. Okay, so what is the difference now between all the different technologies we talked and say metmax, permeabilized hepatocytes, intact, which are the generally utilized hepatocytes across, then microsomes. Of course, we'll cover in detail later on about microsomes. Even we'll discuss how actually microsomes are made, and then S9 fractions. So when this any of this thing you can use for metabolism. So if you endoplasmic reticulum, do you see that in MetMax hepatocyte? Yes, 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 yes. So it is there everywhere, and this is most important because all the isozymes which we are talking about, they come from endoplasmic reticulum. Cytosol in hepatocytes, yes, but microsome, no, because we actually the process you will see that there is a lot of we just do the homogenization, we do a lot of centrifugation, so we really disturb the cells. Okay, and this is taken from the supernatant, the cytosol still is present in S9. Mitochondria again in hepatocytes, yes, but it's not present in microsomes in S9. So as you can see, lysosome, Golgi bodies, plasma membrane, nucleus, all of these are missing in microsomes and S9 fraction, whereas they are present in MatMax as well as intact hepatocytes. How does they vary in terms of their usage? So let's understand that when you have a MatMax hepatocytes, they can be stored at -80. You see, whereas intact hepatocytes, by always you need to have a liquid nitrogen, okay, because they are in cryopreserved form. But this can be preserved at -80, so that's the biggest advantage. So is the case with microsomes and S9 fraction. You should have a storage condition for -80. So MetMax hepatocytes are easy to use, just like microsomes and S9. Hepatocytes, you need a liquid nitrogen. Here, centrifugation required? No. You can without centrifugation, you can simply use it. Hepatocytes, you may have to do microscopic examination. Hepatocytes, you can do. Metmax, you can't do because anyway we have disturbed the cell and they are made porous. So now you don't have to do microscopic examination. Same is the case with microsomes and S9. We just look at the quantitative analysis, that is disappearance of parent compound. That's all we look for. Cell counting, again, only in hepatocytes. Cofactor addition, it's not required in this too. In microsomes and in S9, we have to use this because we have to add NADPH. So without NADPH, your reaction may not start, right? In here, we already added, so you don't have to add anything additional here. Intact, anyway, you don't need because it's already present in there. Thaw and use, so yes, you can just thaw and use MatMax, but not is the case with hepatocytes because you need a recovery medium. Here in microsomes and all, you need to add NADPH to it. So from ease of using, it looks like MatMax is very easy to use. Okay, and this is one example of cryopreserved human intestinal mucosa. 3A4 activity has been studied. So you can see almost all the herbal plants have been taken, and we just checked herbal supplements. Do they really do the inhibition of 3A4 enzymes? And you'll be surprised. Of course, green tea and all have very little potential. So that's why we say they are good because they don't have any other side effect. In fact, they add good for antioxidant addition, but they don't really do inhibition. So that's good. Whereas many of this herbal stuff which we tested, all of them are showing inhibition potential. Okay, this data was generated within India, I guess. So, application of in vitro enteric systems for metabolism and for drug-drug reaction, right? So we can generally say now that you can do a study in animal hepatocytes initially, right? Do it in human because you can do practically at the same time. You don't need any additional permission to do that. So you will have a species comparison. From animal hepatocytes, you can interpolate and you can check which is a better species to do the in vivo testing in animals, right? And once you have animal data here, once you have human hepatocyte data here, we can use actually modeling, and then we can predict what could be the fate of your drug when you test in vivo, in human. You got the point? So we start with animal hepatocytes, we do it in human hepatocytes, do the comparison. From animal hepatocytes, we can extrapolate which ideal species for animal in vivo testing. From that animal data, in vivo data, and human hepatocytes, we can always predict what is going to be the hepatic drug properties in human, in when we actually dose them in human, right? And once from hepatocytes in vitro to in vivo, you try to extrapolate. Uh, PBPK modeling can be done, right? And of course, we have to keep in mind the physiological factors, genetic polymorphism, all those things will play a role for how accurately you can predict. As we already discussed, even up to prediction of 0.6% is more than sufficient, at least to get an idea about how our drug is going to behave in human in terms of metabolism. So with this today's discussion, what we have discussed, that yes, you can use cryopreserved intestinal mucosa, right? That up till now, this was the state. Everybody used to concentrate only on hepatocytes, but here onwards, I would wish that if you are into drug metabolism, pharmacokinetics, and if you are doing research in a relevant area in your college, in your university, or in your job, please start considering cryopreserved intestinal mucosa also, because intestinal mucosa will see the drug first even before liver. And if your drug is substrate to any of the metabolism pathways or for the enzymes which are expressed in intestine like 3A4 or other isozymes, then metabolism can happen there itself. So now we say that you do animal cryopreserved intestinal mucosa study, human cryopreserved intestinal mucosa study. From this, do an animal test, animal enteric drug properties. You can in vivo actually do it or predict also. From this data and human intestinal mucosa data, you can find out human enteric drug properties. So once we go in human, we call it as first in human. We will know, we'll have fair understanding about the enteric drug properties. I mean, the metabolism perspective, how it's going to be in intestine and how it is going to be in liver when we go for human first in human trials. Okay, so with that, I think we have completed about the cells. Let's just quickly continue our discussion and jump on to in vitro ADME assays. Okay, so today we will talk about in vitro absorption, distribution, metabolism assays. We'll try and cover as much as we can, and then we can stop and we can take a few questions. Okay, so we have started our discussion from introduction to drug discovery and development. Then we moved on to alternatives to animal experimentations, talked about hepatocyte, enterocyte, few of the other technologies, and now in continuation we will talk about in vitro assays as well. Okay, so again, this we have covered in detail, but I will just spend 1 minute on it to make all of you understand the role of ADME. Okay, once you take a drug orally, again same example, if you take paracetamol, cetirizine, anti-inflammatory drug, practically any drug orally, it reaches the gut, that is intestine. Through portal vein, it will go to liver. In liver, it can be either in free form or it can be bound to the protein in the blood itself. The free form of the drug is responsible for site of action. It binds with the receptor and will elicit a pharmacological action, or it can go to tissues and will be there as a tissue reservoir. In tissues, it will be bound to proteins because tissues are also proteins. So that will be non-specific binding. And once the bound drug in plasma, that is plasma protein binding. So even those studies can be done. The free drug can, after liver, after the metabolism, it can go through kidneys and then it can excrete out of the body. From liver, after metabolism, now the metabolites are considered to be more polar compared to the parent drug, so they are easily filtered through the kidney and are excreted. So this is the whole pathway when any drug is taken orally. What happens when an intramuscular injection is taken? So as you can see here, directly drug is going to the tissue reservoir, right? Because we are giving in muscles. So from tissue, then through the blood, it reaches the circulation. The free drug again responsible for eliciting pharmacological action, and the bound drug will anyway stay in bound form and slowly it will be released, okay, over a period of time. When through the circulation it reaches to liver, again undergoes metabolism. Metabolites then will become polar. Through kidney, it will be excreted. Yeah. And third form of administration of drug is intravenous. As we see that many times when somebody is really feeling very weak, or they are not feeling, I mean, they're completely energyless. People, doctor have to give uh, intravenous glucose, dextrose, right? So immediately they feel energetic. Why? Because it is directly delivered in your circulation system, right? Circulatory system. So once intravenous drug, it comes into circulation. Again, free drug responsible for action. Remaining drug can also travel through blood to different tissues, will be bound there. Within blood, in plasma, proteins, it will be bound to the proteins. And through circulation, once it reaches the liver, again it undergoes metabolism and metabolites will be excreted. So finally, through kidneys, that excretion happens. Sometimes, I'm not, of course, in this figure it is not mentioned, but sometimes even through bile also they are coming out. They are eliminated, excreted through bile also from gallbladder. Okay, so after liver, it goes to bile. Through gallbladder, from bile it can be excreted. So that is also one way of excretion of the drug. So this is the fate of the drug after oral, intramuscular, and intravenous dosage. And of course, one thing I didn't say, whatever drug you have taken orally, which is not absorbed, it is in unabsorbed form, will come out of your body again excreted in form of feces. So this is the fate of your drug. So let's understand the first part, absorption, as I said, which is the first step. From esophagus, it reaches the stomach, reaches the intestine, and absorption can happen anywhere. So that absorption, what are the parameters? So fraction of administered dose and rate of absorption. Of course, based on rate of absorption, you can make out how much drug is going to be absorbed. So are there any factors which really influence the absorption of the drug? So answer is yes. What are those factors? The lipid solubility. I already told you in intestine, it's a lipid bilayer membrane. So lipid solubility will really help in permeability of that drug. So it will help in absorption. Molecular weight, of course, smaller molecular weight versus larger molecular weight. The permeability really depends on that. Polarity, again, very important. Route of administration, as you can clearly understand, orally it has to see few organs before it really gets absorbed. Intramuscularly, through muscle it comes through, and intravenous, there is no question because directly it is delivered in circulatory system. So distribution. So after absorption, the drug gets distributed across body in different tissues. So let's see, it is governed by what? So again, lipid solubility helps a lot. Ionization helps. We are talking about binding, plasma protein binding. That will also decide the distribution, right? Difference in regional blood flow, that is also very important because different organs have different blood flow. So the organs which are heavily populated with blood flow, vasculature, there more blood will reach, so there will be more delivery of the drug compared to the organs where the blood supply is less. There will be less supply of the drug, okay, distribution of the drug. And disease of liver and kidney. So all of you know that we always say that in elderly people there will be they have to be careful when they prescribe the dose drug. Doctors needs to be careful for the elderly people, and they always ask you, right, that do you have any disease? So the reason is, if at least up till now, based on our discussion, all of you have made out and learned that metabolism happens in liver. So if your disease is related to liver, suppose it's any fibrosis or grade 1 fatty liver or a grade 2 fatty liver. So liver is affected means the enzymes responsible for metabolism in liver would also be affected, right? They could be underexpressed, they could be overexpressed. So based on that, the fate of the drug would vary. So that's why it should be known to us if at all a patient is suffering from liver disease before prescribing any drug. Kidney, why? Because as we have seen, no matter what administration of your route or administration route of your drug is, final excretion is happening via kidneys, right? So if there is a kidney disease, so then there is a less potential of individual to excrete the drug via kidneys. So there are chances of toxicity if you give them drug, more and more drug. So that's why doctors will always ask you, is there any liver or kidney disease? And based on that, they can really decide how much dose needs to be given. Metabolism, again, what is metabolism? Basically, see, metabolism is nothing but biotransformation of drug in body. Of course, it's a natural process to convert non-polar liquid soluble compounds to polar lipid soluble compounds. So you see, that's what basically metabolism is nothing but it's making your molecule bulky. Somehow it is making it more polar. Why? Because if they are polar, they can easily excrete through kidneys. So that's the whole purpose. So this is all natural mechanisms. I mean, nobody has really done anything to activate that. It's already there. If your liver is functional, it is always happening. Any drug you eat, anything you eat, any Ayurveda medicines you eat, eventually through liver, metabolism has to happen. So you see, this is how, although we say we have huge understanding of science as on date, but still, if you think from a spiritual perspective about our body makeup, where we have no contribution, right? And then it is so perfect. You see, everything is well defined. So even in liver, beautifully this all mechanisms are defined. Phase 1 metabolism, phase 2 metabolism, be it oxidation, reduction, simple reactions, and if your molecule can be made polar, or by adding some moieties, glucuronide, so glucuronidation we call it as sulfation, and all such mechanisms. So you see, body itself has all the mechanisms in place. Unless you really do intervention in any of those, I think things will really work fine. So that is metabolism. But as a student, for you, for understanding, there are 3 4 areas. Just with example, I tried to put it here. Like you see, there is conversion of an active drug into an inactive metabolite. A normal scenario where a drug is active, but after liver, after metabolism, now it becomes inactive because metabolites are inactive. So like brufen, paracetamol, this is what is happening. They are active drugs, but in liver, they're converted into inactive metabolites. Then there is something where you take an active drug and it can still be converted into an active product. Means a metabolite can also be an active product. So say phenacetin you take, which is itself is active, and once it goes to your liver after metabolism, acetaminophen, which is again having same effect. So your metabolite is also active. Now third way is you give your drug as an inactive form, okay, and in body it will convert into a active metabolite. So that is also happening through liver. So there is a concept of prodrug. Like we will see this when we talk about solubility later on. Sometimes or many times it so happens that our drug is actually is not getting solubilized, or more so there is a permeability issue. It cannot pass through blood intestinal barrier by lipid membrane. So what is the way forward? So those drugs, people then mask that drug into something else, and now it becomes a prodrug. So with this masking of the properties of the original drug, you can actually use nanoparticle technology or everything is prevalent nowadays. So what happens is, now with this improved moiety, the permeability is improved. But as soon as it is permeable, it reaches to liver, it can really now release the main drug, and that becomes an active drug. Okay, so prodrugs, again, by adding some groups to your chemical moiety, you can make a prodrug. It can be an inactive drug, but permeability, if it is improving, why can't we do it? And in liver, if that addition of the moieties which you have done, if that gets cleaved, now your drug is released. So that factor of permeability issue has been sorted out, and now your active drug is circulating in the blood. So that is called prodrug effect. So prednisone to prednisolone, like prodrug, there are so many examples. And then no toxic or less toxic drug or toxic metabolites. This is also very important. Sometimes our drug is converted into a reactive metabolite, and that is not a acceptable phenomena, because the reactive metabolism or reactive metabolites, they actually can lead to mutagenicity. They are teratogenic, and many times they are hepatotoxic also. So if your drug is having a potential to form a reactive metabolite, then you should know very early in drug discovery, right? That's why we do this metabolism studies early, so that all this can be captured very early. So once your drug is having a potential to form a reactive metabolite, we can just stop the drug there itself, or we can do some modification in the structure of your drug, and somehow we can avoid the formation of reactive metabolites, so that your molecule, which is otherwise very highly potent drug, can now go further in drug discovery and development. Okay, so this is about biotransformation. As I was talking, oxidation, reduction, hydrolysis, cyclization, decyclization, all these simple reactions in order to make your drug polar. If it's not possible, then liver will go do the phase 2 reactions, where they will add the glucuronide. It's called glucuronide conjugation, acetylation, sulfate conjugation, methylation. All these things are phase 2 reactions in order to make your molecule more bulky and thereby more polar and easy excretion. Yeah. And again, factors which can affect your biotransformation, as you can imagine, concurrent use of drugs. We will talk about it in detail going forward, but just very quickly, suppose I'm taking metformin and I'm taking paracetamol. Now, two drugs are both going in my body because I was having fever. I'm also having diabetes, so I'm taking this drug as well. In case paracetamol is responsible for CYP3A4 inhibition, and 3A4 is responsible for mechanism of metformin, then what will happen? Now, although I've taken same dose which I'm taking daily, but just because of fever, I started taking paracetamol. Now the 3A4 expression is inhibited, which was responsible for metabolism of metformin, which is an anti-diabetic drug I was taking. So what will happen? More and more drug will get accumulated. So excretion will be slow, and more drug will be accumulated, because I'll be taking dose every 6 hours. So eventually, if at all there are any toxicities because of drug accumulation, it may happen, and I will never understand why it's happening. But maybe because I started taking paracetamol, I've taken morning, evening for 3-4 days. This is a momentary inhibition of my cytochrome P450 enzymes which has happened. So that's why concurrent use of drugs can really impact the biotransformation. Genetic polymorphism, yes, there is a term called fast metabolizer, slow metabolizer, or medium metabolizer. So that's based on the genetic expression of the enzyme. So polymorphism, all the biotechnology and pharmacy student will know that it is the expression of same gene, or in a different form in different individuals, right? So that is polymorphism. So that can also affect. So sometimes drug is toxic to few individuals in the entire population. Why? Because particular enzymes could be less expressed, which is responsible for metabolism of the drug they're taking, which is expressed normally in all the other individuals, say for 3A, for example, and whereas in that particular individual, for some reason, 3A4 is not really expressed in the same way. So any drug which is getting metabolized by 3A4, if it is consumed by that individual, there could be a chances of some adverse effect because of accumulation of drug. So that is genetic polymorphism, right? Age also really affects. Coming to excretion, again, this is just the theory that yes, excretion happens through urine majorly, but we should also know that through bile, through biliary excretion, sweat, saliva, milk, pulmonary, these are all the routes of excretion. So, what have we studied so far? So that is safe, tolerable levels of exposure, dose, dosage regimen. We will cover about all this thing, but what we have understood right now is about ADME general principles, right? What is absorption? What are the factors responsible for that? What is distribution? What is metabolism? And what is excretion? Yeah. Later on, when we get into discussion of pharmacokinetics studies, I would like to talk about this, all the parameters. Let's see if time permits, we can talk today or we'll in subsequent classes. That whenever we dose a drug to any individual or to animal, we collect blood out of the animal at different time points or from human, and then we either isolate segregate plasma from that or serum, and then we try to quantify the drug. So what happens is, suppose you take a paracetamol, okay, after 5 minutes we draw blood from you, after 15 minutes we take another sample, after 1 hour, 2 hour, 4 hour, 8 hour, like that different time points we start collecting blood from you after you've taken paracetamol, and then we centrifuge this blood so that we can separate the plasma, and then that plasma we put it in -80 till further analysis. But whenever I want to quantify, I'll take out that, I'll thaw that, and we'll talk about bioanalysis, how to handle the biological matrix. But just in a nutshell, I'll try to quantify how much of paracetamol is available in my body after 5 minutes from that sample, after 15 minutes, after 1 hour, after 2 hours. So it will give me a complete profile. When I plot a graph between concentration and different time points, I can see the path of the drug in my body. That okay, it was higher at initial time points, then it keeps on, it reaches the plateau, then it starts decreasing, and there will be some point in time where I don't see any concentration. Means the drug has completely cleared from my body. So from that time versus concentration graph, generally across the world, any paper you take, these are the parameters which would be mentioned. Okay, they would report clearance. That clearance of paracetamol is this much. Okay, or clearance of this drug is this much. This would be seen. Half life of my drug is say 6 hours. Somebody says 8 hours. Some drug will have 2 hours. Okay, so what does it mean? Somebody would say that volume of distribution of the drug is this much. And finally, we all talk about this term that, oh, bioavailability is good, bioavailability is bad, bioavailability is low. So we will talk in detail about these parameters. Okay, that when you do a PK study, you can actually find a clearance, half-life, volume of distribution, and eventually, even if you do a BA/BE studies, you will be interested in bioavailability. So we will understand about all these parameters in detail going forward. But in today, let me see through in vitro what all we can cover from aqueous solubility, octanol solubility, plasma, gastrointestinal fluid stability, a simulated one, and microsomal stability. We'll try and cover as much in vitro assay as possible in today's class. Okay. So let's start with aqueous solubility. So aqueous solubility is defined as maximum amount of solute that dissolves in a given volume of solvent, right? Very simple. Maximum amount of a chemical that dissolves in a given amount of solvent is called solubility. So in other words, you keep on adding the chemical into a solution till it completely dissolves and forms a clear solution, till you reach a stage where it further stops dissolving. That is called the aqueous solubility. Okay, so it is very important for drugs. For drug discovery and development, one of the very initial parameter which we study for any given drug candidate under screening is solubility. So that's why we should always have understanding on solubility. We will understand in few slides why the knowledge about solubility is important early in drug discovery. This is just a simple thing I have added. If you add a water to any chemical unknown to you and take a litmus test, If red converts to blue, you will have an idea it is a base. If blue litmus converts to red, means it's an acid. If there is no change in color, it could be neutral. Suppose if it is insoluble, then you try to make it solubilize. We used to do it in college by adding some aqueous sodium hydroxide, and now again we add bicarbonate, and then we see if it's a carboxylic acid or a phenol, so it is a soluble or insoluble. But this was all very rough way of checking the solubility. Earlier we used to do. Okay, here as I'm saying, as I said, it's very important to understand about solubility early. This is the reason, because if you don't have understanding on solubility, you may actually get data from metabolic stability, okay, and you would incorrectly interpret the data. It may have a solubility issue, but since you're not aware about the solubility, you may incorrectly interpret the metabolic stability data or reaction phenotyping or CYP induction or CYP inhibition. Why? Because, say for example, my drug itself is very lowly soluble, okay, and I just made a solution out of it, say 1 mg per ml solution I have made, okay, and I have done a metabolic stability, added enzymes into it, I mean to say microsomes, or put it into hepatocytes. After incubation, I've taken the supernet and quenched my reaction, and again I tried to quantify. Now I started my reaction with 1 mg per ml. After analyzing the supernatant after incubation time, I realized that only say zero point 0.1 mg per ml drug is remaining. So what is my interpretation? That, oh wow, I started with 1 mg, I end up having zero point 0.1 mg, 90% metabolism has happened. But whereas because the solubility was less, even though I added 1 mg, it was never soluble. So it just precipitated. So I would have started my reaction with only zero point 0.1 mg, you see, or even 0.2 mg. So instead of concentrating on solubility issues, I have now taken a decision that, oh, my drug is 90% unstable. Within this incubation period, only 10% parent is remaining. So there is something wrong with my structure. I cannot take this thing further. That could be wrong because solubility itself was very poor. So that's why we say that always do solubility prior. So you will have an idea that, okay, I have added 1 mg, but actual concentration to consider is only 100 microgram or zero point 0.1 mg per ml, right? So solubility, anything less than 1 microgram to start with, actually it's not a good compound. You will have a lot of struggle going forward. Even PAMPA assays, I'll talk about what is PAMPA and what is CACO, but just for your understanding, it is parallel artificial membrane permeability assay. It's a model, cell, not a cell, non-cell based model, basically, where we don't use cells, and artificial model, basically it is to understand the permeability. Same way, this is a cell based model called CACO2, canine adenocarcinoma cell line. So that is also it will be undetected if you take a drug which is very less soluble. Same way, when you do protein binding experiments, this will hurt you. So in a nutshell, to do any in vitro assays or in vivo assays studies, solubility always hurts if it is very less. So that's why you have to counteract solubility at first. Okay. So what is the importance? As we have studied, it is fundamentally important property that affects not only the potential of drug absorption after oral administration, but also to administer it parenterally. Why? So if solubility is good, it will affect the permeability through intestine. But if solubility is not good, just an example, here onwards, I'm sure you must be, all of you must be aware, but please make sure that next time whenever you get to see any injection, you will always see there are always clear solutions. Okay. Why? Because if solubility is good, only then you can form such a clear solution. Correct? Because for giving intravenous, because it's a natural blood circulation going on and you're trying to inject anything, so if there is any undissolved drug inside or anything else, it will precipitate immediately and there could be some reactions happening. So that's why for intravenous administration we always see dextrose, normal saline, sucrose, whatever intravenously is given, you will always see their clear solutions. Okay, so for a parenteral drug, parenteral means systemic circulation. So for parenteral administration, always a clear solution is required, and that is possible if solubility is good. Orally, still it can work, but parenterally solubility has to be there. Okay, so it can be done easily in each one of your labs, in your college, in your university. You can actually do this experiment very easily. So I was talking about in one of the earlier class about BCS class of classification of drugs based on solubility. So there is class 1, class 2, class 3 and class 4. So class 1, high solubility and high permeability, means the compounds which are highly soluble and highly permeable. So our goal, our aim, our wish list for the appropriate optimal drug to start with is always class 1. Okay, that yes, if my drug is highly soluble, if it is highly permeable, at least ADME issues would be very less for my drug. Class 2, those who are having low solubility but they are highly permeable, right? Class 3, those are very highly soluble but low permeable. And then class 4 compounds, they have low solubility and at the same time they have low permeability. And you know what, friends? Unfortunately, in a drug discovery development scenario, more than 75-80% cases, whatever drugs, whatever chemical class we start with for screening, they're always having class 4 classification drugs. I mean to say, they fall under this category. They are low solubility, they are lowly permeable. So your challenges, your struggle starts with this itself, right? As I said, solubility will limit your bioavailability, correct? Even permeability is low, that will also limit your bioavailability of your drug. So bioavailability is nothing but actual concentration of the drug at site of action. It is bioavailability. So as you can understand, it is denoted by fraction F, and it is always given in percentage. So 100% bioavailability means 100% of the administered drug is available at the site of action. But in reality, it's not possible, right? So anything above 50% is also fine, above 75%, above 80% bioavailability is fine. But if your molecule is having low solubility and low permeability, then there'll be a lot of struggle to make it highly bioavailable. Nowadays, actually, there are means and ways where we can increase we can sort out these issues. How? Suppose your drug is in class 2. What you can do? You can reduce the particle size. You can convert your drug into a salt form. You can use some surfactants. Nowadays, nanoparticles you can use, right? Cyclodextrin complexes are being used. Sometimes just by adjusting the pH, uh, you can use emulsification. You can use solid dispersion. So these are all the techniques. Over a period of years, formulation scientists have really come up with all these solutions so that even if your drug is falling in phase class 2 classification, by using any of these techniques, we can still make it highly soluble and highly permeable. Suppose class 3 classification drugs. So what do? We can use efflux inhibitor because permeability is an issue. So I was talking about transporters. If your drug is a substrate to PGP, and now PGP is a transporter which is expressed in the intestinal barrier. So what happens? It will inhibit the entry of your drug inside the intestinal cells, and it will push it back into your intestinal lumen. So what happens? You can use in this case efflux inhibitors. I can use some other drug along with it, which can be a transporter inhibitor, efflux transporter inhibitor. So what will happen? Now my drug can easily pass through. So that's how you can increase the permeability. Then there are capsules, lipid filled capsules. So even this capsules also can help in increasing the permeability across the intestinal barrier. And then GI mobility considerations also, like sometimes they say that please take this drug before food, after food. Why do they say that? Because that can affect the presence of food or absence of food can affect the permeability or absorption of your drug. That's why they may say that this drug has to be taken before meal or after meal. Okay, and class 4. So anyway, class 4 is the best scenario, but there again we can use prodrugs, we can use co-solvents, surfactants, and all this thing, class 4, low solubility, low permeability. So you can use nanoparticles, you can use your drug in salt form. So these are all means and ways how you can deal with a compound which is lowly soluble or lowly permeability, or they have either of these issues. All right. So just remember class 1, 2, 3, 4: high soluble, high permeable, low soluble, high permeable, high solubility, low permeability, and class 4 is both low. So just remember whichever way you want to remember. This is BCS class of classification of drugs based on solubility. So let's jump into types of solubility. So there are two kinds. One is kinetic, another is thermodynamic. Okay, so kinetic solubility is generally what we use in screening assays earlier in drug discovery. So what we do is we by default more than 95% of the chemical materials, they are soluble in DMSO. So we immediately start DMSO stock solution. We prepare very high concentration, and then we start incubating at different temperature, and then we check the solubility, because there will be an incubation and different time intervals. We'll start taking the samples and we'll see. So since this can be done in a plate format, more than 150 compounds per day can be done this way. At different pH also we can check the solubility. Thermodynamic, generally, as you can see, thermodynamic talks about equilibration. So we start with the solid form of drug and we keep on mixing with a solvent over controlled environment condition for longer period of time. So since you need a lot of stuff to do this, right? Because you want mixing, you want control temperature conditions, and there are a lot of manual interventions. So the throughput fashion, it is low. Only 20 compounds a day can be used. So let's talk about how exactly to do this. As I was telling you, it's easy in your lab conditions. You can do. See, quantify test substance necessary just to saturate your desired volume of water is what we are taking, right? So take 5 times that quantity. Suppose you have a rough idea. Take 5 times of that into 3 glass vessels fitted with a glass topper. Okay. Now volume of water chosen in function of analytical method, solubility range. Water is preferred because if anything is water soluble is the best drug, but sometimes it's not many times it's not possible. So whatever solvent, methanol, ethanol, whatever solvent based on your solubility prediction, you can take that, and their vessels are tightly stoppered, and they are mixed up on a shaker at 30 degrees Celsius. After 1 day, because you added in all 3 vessels, after 1 day, 24 hours, vessels is equilibrated for 24 hours. In between, occasional shaking should be there. Okay, and now you can take from the one vessel, you can take after 24 hours, check for the concentration of the substance in the clear phase. They could be, if it is completely solubilized, great, it will be clear. If not, then whatever clear phase you are seeing, take a sample from that. Mass spec, it says mass spec, but I would say HPLC is sufficient because it will be highly concentrated sample. So mass spec may not be required. Just use HPLC. If it is mass spec, think of diluting your sample. Otherwise, a lot of carryover can come. Okay. After 48 hours, take from the second one, and then after 72 hours, take from the third one. So you have 3 equilibration time: 24 hours, 48 hours, 72 hours. So if the concentration measured in at least 2 last vessels do not differ by 15%, means we have reached the equilibration, right? If it keeps on increasing, suppose in 24 after 24 hours, it is showing me concentration 1 mg per ml. After 48 hours, it is showing me 2 mg. So it is increasing. So I have to keep on increasing the equilibration time till 2 readings will show me difference not more than 15%. Means now it has reached the equilibration. So that now gives you the correct solubility data with accurate equilibration, right? Easy to do, no sophisticated instrumentation required. You can very well do it, and you can identify what is the equilibration time for my best solubility of your compound. Okay, let's move on to octanol solubility. We have talked about solubility, but let's talk about octanol. So as octanol, the name suggests one octanol. It is also called as octane 1-ol. It's basically an organic compound, and it's a fatty alcohol. As you can see, OL comes means it's a alcohol. So it's a fatty alcohol. Okay, and It is basically in perfumes, and it's for the Easters. It is used in perfumes and flavors solution. Octanol is used, but the odor is really very pungent. If ever you would have used it, or if you use it in future, you will see it's having very pungent odor, so you can avoid smelling that. But generally, what is said is the fatty alcohol basically it is say it mimics the intestinal barrier, because intestinal barrier, as I mentioned, it is bilipid layer. So somehow it mimics the characteristics of an intestinal barrier in terms of lipid nature, right? So we use water, we use octanol, and we see our drug distribution between water and octanol after certain incubation period, and whatever data we got, that is called octanol solubility, and it is denoted as log P. So log P of your molecule we can find out using octanol solution. Okay, so basically when you use a water and octanol partitioning, it is an approximation of partitioning between cytosol and lipid membranes of the intestine. You got the point? So basically it's again very simple to do. And I was saying log P, how it is defined. So it is a quantity which is independent of concentration. So it is like concentration in your organic layer divide by concentration in the aqueous layer, which is generally water. Organic is your octanol. That will give you the partition coefficient. So log P value. Okay. So how do we do it? Take equal volume of water. Take equal volume of octanol. Okay. In a vial, in an Eppendorf tube, or in a beaker, whichever way you want to take it. Take equal volume. Okay. And then just add your concentration of your compound in water and allow it to mix. Okay. And all right, so let me rephrase it. What exactly you have to do it here is because we are assuming we don't know what is the solubility in octanol. So what we will do is we will take the drug in a water, okay, and we mix it a little bit, and then we add octanol to that, okay, because if starting onwards, if you add both together, then the partition itself will get affected. So initially you have to solubilize your drug in aqueous, and then we are adding the octanol to that. Okay, now you have to mix it up well. Now the mixture is really mixed well, rotating the sealed bottle for 50 times, right? And sometimes we keep it for overnight shaking also, and next day we will try and see, you will clearly see separate layer of octanol and water. Now what you have to do is very carefully you have to take a sample from water side, and then carefully you have to take a water from octanol side, a sample from octanol side also, and then go to mass spec or HPLC and find out the concentration. So very easy to do. You see, only Eppendorf, vials, beaker, or any such thing would be required, where you can weigh your drug. You can have added water, same volume you have to add octanol, mix it up well, put it for shaking on a shaker at a temperature condition if you want, and next day just you take out the different layers and take samples, and then inject it on a mass spec and find out the octanol solubility. Again, these are just predictors, okay, these cannot tell you exactly. So I won't say 100% is going to be reflected when you actually go in live system, but at least it will be able to tell you, rank order the molecules. You may receive 5 different scaffolds from chemistry. Under those 5 different scaffolds for the same project, there could be 25 different variants they would have synthesized with small variations. Now all of them can't be tested in animals or human. So this is how we do slowly in vitro assays, and try and find out that few of them may have issues with solubility. If you can just chemist can work around, or if required we can drop those, right? So that's why we say you can use this in vitro assays for screening of your molecules. Okay, let's go to next. Plasma stability. Again, see, I have mentioned all the assays which are very easy to do at your place, and sometimes once you do few rounds of this experiment, and when you get to industry, and when in interviews or while discussion, scientific discussions. When you share, I think they'll be highly impressed because these are the normal in vitro assays, which is believed that a student who is doing masters or who is doing his PhD in pharmacology or pharmacy related subjects, or even for chemist, he should know about what is this log P, what is solubility. So all these assays understanding would really help you. So and then it's if you actually are working somewhere, then by all means, this experiments will help you to screen your molecules in different projects. So let's talk about plasma stability. So plasma stability, again, as the name suggests, we have to take a drug and we want to check the stability in plasma. So first you need to have a blood from animal or from human, centrifuge it, okay, 5000 RPM for 10 minutes or for 5 minutes. You will see a beautiful layer of plasma is separated. Take out those plasma, and now we will check the stability of our drug in plasma. Okay, so why do we really need to do this plasma stability? Okay, that also I will explain you because you see in terms of enzymes in liver, I talked about CYP450 enzymes which are available in liver now, which are expressed in liver. Even in plasma, there are hydrolytic enzymes. Which are those? Cholinesterase, aldolase, lipase, dehydropeptidase, and alkaline and phosphatase. So what happens is, if your drug, okay, or your prodrug, essentially prodrug, we'll talk about it later, why prodrug, if they are liable to any of these enzymes, what will happen? The moment they come into contact with blood, which is having plasma, and plasma is having all these enzymes, and our drug is substrate to that, then it will cleave that. Yeah, if it's a prodrug concept, and if your concept is that your prodrug should be cleaved in blood, then that's the best scenario. Even then, this assay is useful because before even dosing to animals or human, just in our lab conditions, we can make out whether my prodrug is working. Suppose if I design my prodrug in a way that it has to be cleaved by lipase, then in vitro itself I can find out whether plasma and my drug I'm incubating together. After some time, when I stop the reaction and I do the sample processing and when I inject, is my metabolite or the active drug from the prodrug is released or not, right? If I see high concentration of parents still after incubation, then I'll know that maybe my prodrug is very strong, and it is not cleaved by lipase. So I may have to again go back to chemistry and work around my structure. So you see, that's how it is very important. At the same, this is I have talked in positive way. Sometimes even my active drug can become inactive because of enzyme action. So even though drug is soluble, even though it is permeable, after permeation when it comes into circulation, and if it becomes inactive by hydrolytic enzymes, then also I will see very poor bioavailability, right? And I will always wonder that, oh, solubility was good, permeability was good, metabolism was not an issue. Why am I having low bioavailability? Maybe because in plasma itself drug has been cleaved and now it is inactive, right? So that's why we should study the plasma stability. Okay. So let's talk about from a chemistry perspective. When you look at a structure of any molecule, if at all you see any of this functional groups, ester, amide, carbamate, lactam, lactone, sulfonamide. So any of these groups are there means your drug could be subjected to plasma degradation. Okay. So as a biologist, as a pharmacist, as a DMPK scientist, you should understand this. So when you see such structure, or even for a chemist, or you can go back to a chemist and tell that, look, your structure may be good from efficacy point of view, but in a cell-based or a non-cell-based assay, but I'm seeing these structures, these functional groups in your structure, and maybe they may be subjected to plasma degradation. So I would like to do a plasma stability study first, and I'll let you know if these are cleaved or not. If at all there is degradation in plasma, then maybe chemist can really appreciate that data, and then he can modify his structure, right? So like peptides, peptide mimetics, all these things, they have this issue. So whenever you have such drugs, always go for plasma stability experiment early. So if you have 5 different groups you are working on, I mean 5 different structure scaffolds, then all of those who are having plasma stability issues, they can be deprioritized, and those who can pass through, they can be taken further. Yeah. So importance, of course, important role in drug discovery development. If our drug would be subjected to degradation or modification by enzyme in plasma, we'll come to know. Majorly, always remember in plasma, what are the enzymes? Hydrolases and esterases. These are the major enzymes expressed in plasma. Okay. So, application, as we have discussed, diagnose the poor in vivo performance. Yes, because as I was telling you, there could be low viability, and the only reason could be because our drug was cleaved in plasma itself, right? If there are any liabilities, if those functional groups liabilities are identified through plasma stability, it will be useful for chemists. They can really modify their structure. And you are working on many different types of structure. At least it will, this information will help your chemist to prioritize the compound which can go further in vivo testing, and which can be which can go back to the chemistry for further modification of structure, right? As I was saying, prioritize synthetic efforts. Screening of prodrugs, this is very important. Before even going into in vivo, just by in vitro, you can go back and tell your chemist that their prodrug concept is working or not, right? And structural modification, as I said, whether it is cleaved or it is not cleaved, whether a prodrug is cleaved or not, all this information eventually will help you to do some structural modification by your chemist of the project. Again, very simple assay to do. How do you do it? We actually dilute the plasma. We can do it directly in plasma also, but I've seen many places, articles, and many labs, they dilute plasma with 80% phosphate buffer saline, right, PBS, at 7.4 pH, and we use incubation condition of 37 degrees. So what happens is, you have this thing ready, 7.4 pH, diluted plasma at 37 degree. You just add your test compound of, say, 200 micromolar. Here it is taken, but you can take, if mass spec is what you're going to utilize as a detector for analysis, then even lower concentration can help. Less than 100 micromolar or a 10 micromolar is also fine. Suppose I want to do quantification using HPLC, then maybe higher concentration, 100 micromolar or 200 micromolar solution of my drug, I need to put it into plasma. Okay, now put it in a shaking water bath, 37 degree. Always do it in triplicates, so that any error while doing transferring this, or while doing pipetting, any errors are there, you can easily find out. So we always prefer to do in triplicates. Now you can take out the samples, because it's a long incubation at 0 minute, 15, 30, 45, 60, 90 minutes. So at different time points of incubation, I can keep on taking my samples. As soon as I take the sample, to stop the reaction, I have to quench the reaction. So I have to stop the reaction. So how do I stop that? Using organic solvent, either acetonitrile or methanol. I would prefer acetonitrile, and ice cold condition, if you use, that would be fantastic. When we talk about biological matrix, one of the technique of sample processing is protein precipitation, right? So one is protein precipitation for biological matrix sampling, sample processing. Another is liquid extraction, and third is solid phase extraction. So we prefer to do protein precipitation here in this particular case, because basically plasma proteins are there, and we want to denature the protein so that your drug can be released. So what do? We do denaturation of protein by adding acetonitrile. And from our experience and doing many experiments, we know that when you use an ice cold acetonitrile, it does better denaturation than using a normal room temperature acetonitrile. Okay, so now these samples are now subjected to centrifugation. So you centrifuge it 14,000 RPM for 15 minutes, preferably at lower temperature. Why at 4 degree? Because we don't know the stability of your compound. So it's better to maintain at 4 degree. And now you have a clear supernatant. And suppose you have taken 10 micromolar, then inject that into mass spec. If you have taken 100 or 200 micromolar, you can do it by HPLC. And now I'll have different time points. I'll have different time point concentrations. So you can easily make out that at 0 minute, before actually starting the reaction, my concentration was 200 micromolar. By the end of 90 minutes, my concentration has come down to 100 micromolar. So half-life is roughly 90 minutes, right? Suppose within 30 minutes, from 200 micromolar, I have come down to 100 micromolar concentration. So half-life is 30 minutes. Or suppose in 5 minutes itself, the drug has it has become from 100 to just 10 micromolar. So more than 90% is cleaved within 5 minutes the moment it has seen the hydrolyzers and esterasers. So you see, this gives you very important information about the potential probable fate of your molecule when it will actually be injected into a live system. So that's why plasma stability, one of the very important assay to do early. So let's talk about stability of compounds in gastric and intestinal fluid. So as I said, when you take a oral pill, say paracetamol or a brufen, any tablet we take through mouth, we take, it goes to esophagus and it is delivered first in stomach where it is subjected to acidic conditions, and then it goes to intestine where more or less not basic but neutral conditions it will be subjected to. So again, how about really testing this in vitro rather than actually going for an in vivo study to really understand what is happening in gastric fluid or in intestinal fluid? We can simply do this experiment in our lab, right? Let's see how do we do it. So first, why it is important? Because for two things, always remember, one is pH, because in acidic pH stomach, pH conditions are acidic, so it will be 1.2 pH at the most. When it goes to intestine, it could be 6.8 plus pH, right? So there are two different pH conditions our drug is going to be encountered with. At the same time, there could be some enzymes in stomach separately, and in liver it could be different. So that treatment is also different. So you see, so the chemical stability of your molecule in these conditions needs to be understood. So very simple assay to do. We call it as simulated because actually we are not collecting the real gastric fluid or a real intestinal fluid. We are just artificially we are trying to create the same environment in a lab condition. How do we do it? Use 0.2 gram of sodium chloride. It is added in 100 ml flask and is dissolved in 50 ml water. We adjust the pH to 1.2. Use any acid to adjust the pH, and then we add 0.32 gram of pepsine. Very important because pepsine is also there in stomach. So we commercially it is available from Sigma Eldridge. You can use pepsine, and then after gentle shaking, you can make up the volume to 100 ml. Now your simulated gastric fluid is ready. Suppose you are using a intestinal fluid, then use 0.68 gram of monobasic potassium phosphate. Okay, and then we adjust the pH to 6.8. All right, and there in intestine, instead of pepsine, we always find pancreatine. So again, commercially pancreatine is available. Again, by Sigma Eldridge, you can easily find this. So you see, just by using normal chemical which is available in any chemical lab or any pharmacology lab, you can actually mimic simulated gastric fluid, and you can make simulated intestinal fluid, right? So once you have made this thing, now you actually add your sample to that, right? And then you can again in triplicates and at different time points keep taking out the samples and look at the concentration. Same way, either in HPLC or you can check in mass spec. I would prefer HPLC because this is not that clean a sample, right? We are adding buffers, we are adding, I mean, not buffers actually, but yes, we are adding monobasic salts and all. So I would prefer HPLC would be a better choice for this because concentration also can be kept higher. Okay, so I think, okay, let me finish this also. So at least microsomal stability will also be over. We still have time. So let's move on to next in vitro assay, which is called as microsomal preparation and stability assay. All right. So microsomes, as we have discussed along with hepatocytes, these are subcellular fractions, right? They are mainly derived from where? From endoplasmic reticulum, okay? And they are rich source of drug metabolizing enzymes. So whatever CYP450 enzymes we have talked about, flavin monooxygenases, and glucuronide transferases, all of them are majorly expressed in microsomes. Okay, so again, we have to do a lot of homogenization while we actually prepare microsomes in lab. So that's why they are having. I mean, you cannot just check them under electron microscope and all, because we are going to disturb all the cells, and we are going to just homogenize that basically. So yes, while making that process from the supernatant, you can get S9 fraction. There you can still find, but for microsomes, we are just looking for plain enzymes levels and a protein level. It needs a catalyst, cofactor in form of NADPH, and that's it. We incubate our drug with that different time points, quench the reaction by adding acetonitrile, and subject the sample to analysis. It's pretty straightforward. Okay, you can make this also in your lab. All you need is rat or mice, whichever species you want to make microsomes from. You need a homogenizing buffer, you need a pyrophosphate buffer, and you need a microsome buffer. Okay, and few surgical scissors, handheld blender or a homogenizer, or you can do it in even a mortar and pestle. And then, as I said, you actually need NADPH to really do the reaction while you do the microsomal stability. So you can divide this into three things. First, you have to actually prepare the tissue, which is liver. You have to isolate the microsomes and actually do the whole process, and then you have to do the stability assays. Okay. So what you have to do is, generally rat and mice, all the pharmacology students at least who would have handled them, or you must be having animal house in your lab in your college. So you have to take this rat, make them cervical dislocation or decapitation or CO2 asphyxiation, and sacrifice the animal. Okay, and then remove the liver rapidly. This is also one way, but this is a published way, and many people do like that. But I generally, of course, before even removing the liver, I do the perfusion of the liver while it is intact there. Okay, so once you perfuse the liver, what happens is it will make it devoid of all the blood supply, so that the dark tan color liver now becomes pale. And here people, what they have recommended is, they take out the liver, put it in ice cold homogenizing buffer on ice bath. Now perfuse the liver by inserting the syringe into the liver vasculature. This is also a way, but you will have to, so many times you have to inject. But while it is intact, there is an aorta which passes through the liver. There you can just put it this syringe, and with one go you can really completely make it perfused. Anyway, whichever way do you do it, it is acceptable. Now excess moisture, by using blotting paper, you can just make it dry. Now weigh the tissue. Okay, now add 3 volumes of homogenizing buffer, as we have mentioned how to prepare that, which buffer we use. Now mince it into small pieces, right? Surgical scissor. Generally we take 4 grams in each of the separate vials to make the microsomes. Further, now those bigger pieces, use homogenizer, handheld or a normal stainless steel homogenizer, 3 to 5 seconds, not more, otherwise it will be completely destroyed. Just 3 to, very quick, and that is sufficient. 3 to 5 seconds, it will just blend it. You will have some kind of a frothy thing formed. Okay. Uh, so what happens? I mean, not much froth should come, of course, but once you use this 2-3 times, I think you will learn that technique, so that frothiness can be reduced definitely. Otherwise, it will cause a denaturation of the enzymes. Okay, and then centrifuge this homogenate for 15 minutes at 12,500 G for 4 degrees Celsius. This is important because the entire preparation of microsome has to happen under cold conditions. So whatever centrifugation steps we will do, we'll use it for 4 degrees Celsius. Okay. Now decant the supernatant into the 1.5 ml ultracentrifuge tube, and now discard the pellet. Okay. Now to that, again you have to add homogenizing buffer at 7.4 pH. Now this time you have to do 1 lakh G. So you see, normal centrifuge, tabletop centrifuge may not go at 1 lakh G. You need ground mounted big centrifuge which can go up to the range of 1 lakh G at 4 degree for 70 minutes. Now decant the supernatant after that, and then suspend has to be resuspended in pyrophosphate buffer. Okay, and then again go for centrifugation for 1 lakh G, this time for 45 minutes. Now decant the supernatant, and that 6 ml microsomal buffer, I mean whatever is there, you have to resuspend that pallet, and your microsomes are ready. Only one thing is remaining, that is you have to find out what is the protein content of that. So during my time we used to use Hartree-Lorentz method of biochemical method of estimation using a spectrophotometric reading. We used to find out what is the protein content, right? Because a specific protein content needs to be used by using microsomes. And until your further usage, you can always keep it in minus 80-degree freezer. As and when you want to do metabolic stability assay, you have to just take it out. Okay. So when actually you want to do it, you will have actually the microsomes up to 0.3 to 10 mg per ml protein you have to take. Your substrate, if HPLC we are using as our quantification technique going forward, you have to use 1 millimole concentration of your substrate. Yeah, and MgCl2 you can add, and you can add NADPH2. Okay, NADPH will actually initiate the reaction. So in total 0.5 ml of potassium phosphate buffer, you have to add all this thing and initiate the reaction within say 5 minutes, 15 minutes, 30 minutes, different incubation times. So after 5 minutes, I'll take 50 microliter. Same process. To quench the reaction, you can use ice cold methanol or acetonitrile. Okay, 100 microliter. And now centrifuge it, inject the supernatant into mass spec or HPLC. And these are a few formulas which you can actually use to calculate the actually half-life. As you can see, 0.693 by K, which is a constant elimination rate constant. You can easily get this actually. Volume of incubation divided by protein in the incubation, and then you can, from that you can find intrinsic clearance, which is nothing but volume, which is volume of incubation, which you know 0.5 ml divided by protein. If it is 0.3, 0.5, whatever, you have to mention that division into 0.693, which is a constant, by half-life. So half-life which you have found, this you can see from the slope and all, based on the concentration versus time graph when you plot. If time permits, going forward we will actually make one graph and show it to you how actually you graph this. You put it on a graph paper, the concentration versus time, and then you can find a slope, and basically you can find out half-life, and that can be utilized here to find out the intrinsic clearance of your molecule. And then you can rank order your molecule, that this is having high intrinsic clearance, this is having low intrinsic clearance. Okay, so that's why you need the prediction. Basically, all this thing will really help you to predict eventually the parameters in higher species or in animals or in human. Okay, so just very quickly to summarize the whole discussion, from the gut wall, solubility, permeability, all this simulated gastric fluid, all this thing can be done in a live system. Through that gut wall, metabolism can happen. Metabolism, because of enterocytes, you can study that, and whatever comes through that, through portal vein, it is delivered to liver, and in liver again metabolism can happen, as we have seen phase 1, phase 2 reactions, and then finally it goes to the systemic circulation, or after metabolism the metabolite will be eliminated from the body. So within the absorption phase is here, liver first pass effect is here, and the first pass before liver happens in enteric cells. So all these factors together will help you understand, gain some understanding on bioavailability. So you see all these experiments, plasma stability, simulated gastric fluid, simulated intestinal fluid, solubility, uh, octanol solubility, or even permeability assays, all this thing will help you understand the ADME properties of your drug. Okay, so eventually it will really help you screen many molecules which you may be working on, and you can prioritize your molecule which will go further in discovery and development, or you can go back to the chemist and actually do some structural modification and to come up with a better molecule. So now you see this many things needs to be done before actually you go for clinical trial. So that's why you need very thorough preclinical trial in place. I hope it help you in understanding that, right? So you see, the release and dissolution happens in intestinal lumen, permeation through intestinal barrier, in intestinal barrier, efflux transporters are also there that will really pull back your drug back into the lumen, and metabolism can also happen. And the second site is metabolism in liver, and the excretion can happen through kidneys and sometimes through bile. Okay, and I think I will stop here, and then we'll take any questions because then now we will, next time when we talk, we will go to discussions on permeability especially. So we have covered in vitro assays like solubility, octanol solubility, log P assays, simulated intestinal fluid and simulated gastric fluid stability, plasma stability, microsomal stability, and now we will jump into next time into permeability assays. Okay, so I'll stop here and would encourage. Would be more than happy to answer any questions if you may have.
Yeah, thank you, sir. Students, anyone having any queries, please put in a chat box. Please respond. Please respond. Anyone having any queries, please respond in the chat box. Students, anyone having any queries, please respond in the chat box.
And
Please.
in fact, even later on, also you can ask your queries. It's not that immediately because you can absorb this whole information, and later on, and these are the emails. So there also you can post your. I'll be more than happy to answer those. But in this forum, if you ask, it would be useful for everyone. So if you ask one question and we discuss that, all the participants can learn from that. So that's the only reason. But I'm open to clarifying your queries anytime during now or even later on. Also, you can reach out to me. Okay. So
Sir, can you explain once again about octanol solubility?
yes. So octanol solubility, I would just say that, see, this particular solubility is generally done very early when a chemist is actually coming up with their series of molecules with slight modifications in their main structure. So we just want to rank order the compounds. Do they have any issues? So you see, permeability, when we actually talk tomorrow, you will understand that actually we can do a permeability assay to understand whether a drug is going to have any permeability issues, whether it will be able to pass through the intestinal barrier or not. So there are cell-based model, there are non-cell based artificial models also, we'll talk about it, but that is later on. First time when the chemist has just synthesized the molecules, is there any way we can do some rank order of the compound, that okay, this compound can go for testing first in vitro and this can go for later on? So at least to do that, we want an idea on two things. One is solubility, one is octanol solubility. So octanol solubility is not a, it's an apparent solubility actually, because what we consider is we use octanol and we consider that octanol will act somehow, it will mimic the structure of our intestinal barrier, because it is lipid in nature, right? So it is fatty oil and we are using an aqueous water. So whatever is happening between a lipid and a cytosolic membrane, this is going to be mimicked completely. It's a assumption. Okay, in reality, there are lot many factors like transporters, pgp, efflux transporter expression, all those things are responsible, but this is very early. So a very simple assay to understand if at all my drug is soluble in lipid layer or not, it will just give you that idea. So you're done solubility, you're done octanol solubility. So it will now give you some confidence that yes, my drug may get through the permeable, it may get through the intestinal barrier, right? So that's why we do octanol solubility. How do we do it? Very simple assay to do. Take your drug in water, okay, some concentration we can take it, or you can use even some other solvents also, but we generally say for aqueous and lipid, so we always say use water. In that, now add equal amount. Suppose I have taken 0.5 ml of water, then add 0.5 ml of octanol into it. Now mix it up well. So the concept here is, if my drug was soluble in aqueous, now is it really getting into octanol? Is it really solubilizing in octanol or not? So since octanol is fatty oil, I consider it as okay, if my some amount of drug is soluble in that, means it will be able to pass through intestinal barrier when actually I give the drug orally. You got the point? So I can rank order. Suppose, okay, so let me finish the process. So you add both of them equal concentrations now, mix it up well, put it for overnight shaking. So whatever equilibration has to happen, it has happened. Suppose my drug could migrate from aqueous into the oily part, then I consider that yes, it could be permeable also. So it is just an assumption. It may not be accurate 100%, but as I'm saying, very initially, even before doing other in vitro assays of permeability, it will give you some idea about the lipid solubility of my compound. So that is called octanol solubility.
Yeah, thank you, sir. What is silicone methods?
What is?
Water in
Okay,
silicone.
okay. Yeah, so basically it is called as in silico method. So you see, up till now, today, what I have talked is basically I talked about in vitro assays. So you are actually doing the experiment, right? Be it a chemical experiment or being it a biological experiment, you're using gastrointestinal fluid, gastric fluid, intestinal fluid. These are actually you're taking chemicals and solutions and reagents and pH adjustment. You are doing it. So these are called in vitro assays. We are not doing it in human. Sorry, we are not doing it in a live system. If we do in live, which we'll talk later, is called in vivo. Now, even before I do this, the first step in drug discovery nowadays is in silico. So in silico, what happens is there are libraries of compounds available, and suppose for solubility, as I said, BCS classification is also known. So there are libraries of compounds available. I can screen my compound. I can give a search that okay, is my drug this particular structure which a chemist has designed. At least he is not synthesized. He has just designed a scaffold. Can this structure have any solubility issues? Can this structure have permeability issues? Are there any known metabolic liabilities with my structure? All these things you can actually do the screening by in silico methods using computer softwares. So there is millions of data already available. Scaffold data is available. The molecular ligand structures, receptors, their 3D protein structures are available. So even the binding of my molecules I can study in silico without synthesizing a molecule. So all this thing is called in silico. So you can actually do you just using computer simulations and softwares, you can find out the liabilities to your molecules, be it ADME liabilities, be it efficacy liabilities, or be it toxicology liabilities. So all these studies using computer softwares without practically doing anything in lab is called in silico, and it is a good way, a starting point. But as I said, whatever is done in silico, finally you have to do in vitro. But yes, in using computer softwares, programs and all, I can screen hundreds of molecules together, and I may find a hit that okay, out of 100 different scaffolds I have tested, only two are showing good activity. So now those two I can take in vitro assays. So you see, first you do in silico screening, then you do in vitro, and then we do in vivo screening in animals, and then we finally go to human trials. So that's the chronology which is being followed.
Yeah, thank you, sir. Sir, one more question, sir. Protein building
Okay,
blinding on
so
plasma stability.
Yeah, so we haven't covered protein binding today. I'll talk about it in a subsequent class. So yes, protein binding is basically what you need to do as one of the assays. Now your question is, will this protein binding affect the plasma stability? So you see, enzymes, as I said, are already available in plasma, in blood, hydrolases, esterases. So what happens is the free form of the drug, okay, basically the free form of the drug is available, that would be exposed to that drug. The bound form may not be, right? So please remember that protein binding, we have to do separately from a plasma stability. We cannot do a protein binding assay. I'll tell you why. There are different principles for that, because we have to do some equilibration and filtration, and those things we have to do. I'll talk about it when we cover plasma protein binding. But in a plasma stability, don't worry about it, because your incubation is very limited. It is not happening in live system. It is in a plasma which we have collected outside. So basically, it is an in situ model where we are adding our drug to the plasma. In plasma, there is abundant supply of hydrolases and other enzymes. In that, if it comes in contact the free drug, it will really do the stability of the data. But plasma protein binding effect won't be there, because plasma stability assays are pretty quick. And eventually, once I denature the protein, all the drug would be released. You got my point? At the end of it, how do I stop the reaction? I just quench it. So once I quench using denaturation using acetonitrile, my all the bound drug should be released. So basically, it will not affect there.
Yeah, thank you, sir. Students, anyone having any queries, please put in a chat box. Conjugated drugs should not be taken at the same time.
Okay, so it is it's a good question. Actually, you see, I'll tell you, as a process, as a process. So when we do the clinical trials, also BAB studies are also happening. But at the same time, you have to see the interaction with concomitant supplied drugs. So that data is also required. Means I have to submit a data where over-the-counter drugs, say you are giving anti-diabetic drug, but before actually regulatory agency gives you a permission to launch that drug, you should submit the data, the interaction with concomitant drugs or with a over-the-counter drug. So what is over-the-counter? Which are anyway available, like brufen or your headache drugs, or for fever, your cold. These are the normal drugs which are taken. So you have to generate a data that there is no interaction happening. And if at all interaction is happening, then you have to put that in your label. And sometimes it is there in the label, right? That they say that many times they say that it is not to be taken in pregnant women. That is just advisory. But sometimes it is mentioned that you cannot take this or consult a doctor before that. This one concomitant drug interaction is taken care initially when we do CYP inhibition studies or CYP induction studies. When we do that, will give you an idea if there is any such potential that with concomitant drug is there any interaction happening or not. But in reality, in real life scenario, do we ever do that? No. Suppose you are taking 3 4 pills, we just take it together, all right? And we don't really care about whether a particular drug will affect the other drug or not. Neither do doctor. Which doctor has ever prescribed us saying that you please take this drug, and then after 1 hour only you should take other drug? Do any doctor say like that? They will simply give you 5 medicines and say that take before meal or after meal. That's the only thing. They are only worried about food interaction. Why? Because that is there in the label of the drug, that with food the absorption is less, or with food absorption is low. So based on that, they can tell you to take before and after, but nobody can tell you that this can interact with other drug, right? But it's a valid point. And as I say, while drug discovery, it can be countered, because while you do a CYP inhibition study or a CYP induction study, we'll come to know that. And even over the counter drug interaction studies also needs to be done. So you need to submit that data. So actually, so it will be known, I guess. But still, sometimes there are some known allergies, or say there are known interactions that even doctors should be aware of, right? So that known interaction should be avoided, but otherwise, by and large, over the counter drugs, that data is always a part of data package which is submitted to ANDA filing. So that you should not worry about.
Yeah, thank you, sir. Thank you so much. Time is up.
All right.
Thank you for your
So
information,
thank you,
valuable
thank you,
information.
thank you, Maheshwari, and thank you everyone for attending this. And this will still continue for next. We have to do this in
On,
for 15. Yeah, for,
on the
till
Monday.
yes. So we will continue this, and feel free to ask any questions. As I already shared my email ID also, you can always write to that, and as and when lectures are over, through chat box also you can publish your questions, and I'll be more than happy to answer. If I know the answer, I will try to answer. If I don't know, I'll go back and find an answer for you. So thanks again. Thank you all of you, and looking forward to connect with you again soon. Thank you. Have a nice day to all of you.
Thank you, sir. Thank you so much. Students, we are having 3 days holidays. Again, we'll join on Monday.
Oh,
Okay,
okay. So thanks, Maheshwari. This I was not aware. So tomorrow, day after tomorrow, and day after, there are no
Sunday.
classes, right?
Yeah,
Oh, okay.
Friday,
So,
Saturday, Sunday, sir.
so class will start on Sunday or Monday?
On Monday, sir.
On Monday, and
Yeah.
tomorrow is Friday, Saturday, Sunday. Perfect. So then, let me take this opportunity, Maheshwari, to wish all our participants, and including you and APSSDC management team and everyone, Merry Christmas in advance,
Thank
and wish you all a very happy holidays. And to all the students, please enjoy this festive season, this holidays with your families, and have a great time with your family and friends. A lot of greetings and blessings and wishes to all of you, and looking forward to connect with all of you on Monday. So till that time, thank you.
thank you, sir. Thank you, students, and wish you the same to you, sir. Merry Christmas. Thank you, sir. Students, tomorrow, day after Friday, Saturday, Sunday, we are having the holidays. On Monday, we will meet the section. Okay. I have given the feedback form. Please submit all of you. Thank you. Thank you, sir. Bye bye.
Thank you. Bye bye. Thank you.
The section is closed.